Welcome to this ESID session on diagnostics in inborn errors of immunity. I am Seppänen and I will be moderating this 1.5 hour session. Thank you and enjoy.
Hello everyone, I'm Garcelon, Director of the Data Science Platform at Imagine Institute. I'm very glad to be invited to present our work in the field of medical informatics. And you can submit during my talk your question via the question answering function in the top right corner of the screen. And hopefully I will try to address your question at the end of the session. So I'm going to talk about artificial intelligence in diagnosing PID, primary immunodeficiency, and also how to accelerate the translational research with the rare diseases data warehouse. About faculty disclosure, I'm co-created with the Imagine Institute, a spin-off of the Institute, CODOC, 3 years ago. So we are in a very specific context with the Imagine Institute because we are based on the Necker Hospital campus. So we are in a very tight collaboration and a very specific translational research context with 34 reference centers for rare diseases and 25 competence centers in Necker Hospital. Let's talk about the data. Hospital data are a mine of knowledge for retrospective studies. It's already well known now for clinical research, epidemiological research, medical economic, etc., etc. But also, we know that it can be very useful to have access to the collective memory to take care of new patients. Jennifer Frankovich wrote a paper in New England on how she used the data warehouse in Stanford Hospital to make a decision on your new patient, very complex patients with systemic lupus erythematosus. There was no information in the guidelines or in the literature, and they used the data warehouse to find patients like her, and they make the decision on the basis of the best data available in the light of experience as guided by intelligence. So we know that we have to use the collective memory, all the data produced by the hospital, for research and for the care of the patients. And the care, it means that we will be able to develop artificial intelligence algorithms to increase the capacity, the ability of reuse this data for the diagnosis. But it's not so easy because this collective memory is quite a mess. All the data produced in the hospital is a patchwork information system with a complex architecture. All the data are compartmentalized with historical databases and several formats and volume and technology. So it's not easy to reuse that, to have access to this collective memory and to reuse it. And if we dig deeper, we have to make a decision about the coded data because most of the time we use coded data like ICD-10 or biological results. But is it enough? There is a case study on autoimmune comorbidities in patients with celiac disease where they try to identify in which kind of data these morbidities can be found. And in this chart, you can see that most of the time, in dark blue, the phenotypes are only in free text and very barely only in coded data. In fact, 81% of the comorbidities are only in free text. So we have hundreds of databases with coded data, but also we have to deal with free text. Because free text is where you have the most detailed phenotypic description, especially for undiagnosed patients with rare disease. Because the clinician can express a doubt of information or absence of signs, and it's basically richer. So our main objective is to use artificial intelligence on all these clinical records to create clinical decision support system for rare diseases in the hospital, especially in primary immunodeficiency. To deal with the heterogeneity of the data, we built a biomedical data warehouse called Dr. Warehouse. And we put in this data warehouse most of the data available in Necker Hospital, structured data, but free text and whatever we could find. We enrich the data with knowledge database, and we address the technical challenge of this integration of heterogeneous data in a single format. And we also deal with all the issues with identity federation, especially with antenatal and postnatal records, which is not so easy. In Necker Hospital, we have 740,000 patients and 7 million clinical reports from 29 sources of data from 1996 to 2020. We have reports of consultation, hospitalization, emergency, imaging, and ATYPICAL, extra. Now we have the data available. We want to address 3 use cases. The first one is to identify the patients that we can use to have a proper cohort to build a decision support system to make a clinical trial. The second one is to extract the information from the narrative reports. And the third one is to develop the clinical decision support system. And one of the objectives is to transform the patient's records into a computable objects for diagnostic support system tool. So we want to transform textual data, all data from a patient, into something that we can use in artificial intelligence. The first use case is to find patients. So the idea is to build a search engine on the collective memory of the hospital to give a proper access to this data in order to accelerate the patient recruitment for clinical trials, for example, to test hypotheses for feasibility studies, like association of science, of drug, and genes, to find patients like mine, like did Jennifer Frankovich, or just to quickly create cohorts, for example, to address artificial intelligence issues. So the idea is to build a Google-like search engine on the patient's records to do this. But if we we put the records in a system like Google, you will have with these 3 queries like Crohn and diabetes, lupus and diarrhea, or NPHP1, we have 350 patients found, and most of them are false positive. In fact, 250 patients are false positive because you have the negation. The patient denies chest pain or doesn't not have diabetes. We can exclude lupus from the diagnosis of the patient. Or the family history, the father has Crohn's disease. And in pediatric hospital, genetic disease, we have a lot of negation and family history information in the narrative records. So we have to develop the algorithm by using artificial intelligence to detect the polarity, negation and affirmation, the experiencer, if it's the family history or the patients, sorry, there is a T, the patients, and the detection of certainty, hypothesis and reality. Maybe the patient has a lupus or he has a lupus. So we applied regular expression methods, firstly, to detect automatically all this information without any annotation of the health report. And now we applied artificial neural network to detect semantic context by manually annotating clinical reports. So we manually annotate, like on the right, the portion of the sentences, if it's negation and affirmation, and he learns how to detect automatically this kind of context information. So we applied those methods on the text of Necker Hospital, and we increased the precision from 30% to 88% with the regular expression, and now we have a precision of 98% with the neural network methods. We have to publish the new results now. It's not the publication is just with the regular expression approach. So we have now a proper search engine in which we can search patients, like in this query, with infection, eczema, and thrombocytopenia. So we can have a tool to create a cohort of patients. Now we want to extract from these patients phenotypes automatically. We don't want to read all the health records, and we want to extract automatically all the information from the narrative records. In order to automate the phenotype typical description of a group of patients, to enrich knowledge databases, for example, or to compute data, those data extracted for artificial intelligence and clinical decision support system. To do this, we combine name entity recognition based on regular expression. Again, if you find exactly the same word in the sentences, it's okay, we'll pick it. And we applied also disambiguation with neural network algorithm. The disambiguation is very useful with words like proteinuria, which can be a lab result or a disease. It depends on the context in the sentence. So again, we annotated 2,000 health reports manually, all the phenotypes, to learn to the algorithm how to detect the phenotype or not. And other colleague in Necker Hospital worked on the detection of drugs and posology in the health reports. So they annotated all the drugs in the health reports to learn how to extract them automatically. For the high-throughput phenotyping, so we applied this algorithm in the data warehouse of Necker Hospital, and for each narrative records, we were able to extract all these clinical concepts. So when we make a query in the data warehouse, like doc8, it will find any patients, and for those patients, we will be able to aggregate all the concepts found for those patients, extracted from those patients. So in this example, we have a description automatically of all the phenotypes associated to doc8 mutated patients. This is an example of the Dr. Warehouse interface. You have the doc8 gene, you have 20 patients, and you have all the 800 phenotypes extracted from the narrative records for those 20 patients and 5,000 health reports. So usually you have to do it manually to look for the phenotypes in the narrative reports. It's automatically done with this algorithm. So we extracted the entity recognition is based on the UMLS metathesaurus, and it includes HPO and OMNIM. And in Necker Hospital, we extracted 33 million of concepts in the medical reports, from the medical reports, and we have 18,000 distant medical concepts. Another example of what we can do, because in the previous slides, you have the concept extracted, the frequency of the patients in the system, and you can also have the median age of the first mention of the concepts of the phenotypes in the narrative reports. It's not really the age of first apparition of the phenotypes, but the first time it's mentioned in the reports. And we did a chart with APDS disease. So we have the phenotypes extracted from the narrative reports on the left, and down in the bottom you have the median age of first mention of the phenotype in the clinical reports. You can see a gradient of apparition of the phenotypes. We published this algorithm in Orphanet Journal of Rare Diseases, and we evaluated the algorithm on 6 cohorts of patients, and we had a quite good coverage with Orphanet, and the difference was due to a lack of concepts in the French concepts in the HPO, not fully translated for now. So we are waiting for the full translation of HPO to increase our coverage of the French vocabulary in our system. And the next step is to build a phenotype-wide association of the extracted phenotypes. A colleague, Antoine Neraz, already built an algorithm, but we have to publish it now and to industrialize the code. The last step is to build a clinical decision support system based on the data integration and all the artificial intelligence already done to transform textual data into structured information. And now we can develop algorithm to try to diagnose patients. So we have a use case. You have a new diagnosed patient with a new gene discovered by a clinician or researcher at Imagine Institute with a very complex phenotype. And we want to find if in all the history of Necker Hospital, we have maybe undiagnosed patients with the same mutation. So instead of using the search engine to find all the phenotypic criteria, we want to compute similarity distance between phenotypic similarity between the index patient, the diagnosed patient, and all the patients of the data warehouse. And we expect that the similar patient may have the same mutation. So we have the index patient, the diagnosed patient, with all the concepts extracted from these narrative reports. And we have the same from all the patients of the data warehouse. And we compute for each patient of the data warehouse a similarity distance with the patient index. And we keep only the top 30 similar patients. The similarity is a vector space model. It's just the cosinus of the angle between the two vectors, the vector of the patient index and the similar patient of the data warehouse. All the concepts are weighted with term frequency, inverse document frequency, that takes into account the rarity of phenotypes in the data warehouse and in the patient. We evaluated our clinical decision support system on 5 cohorts of patients. One was APDS, dystrophic epidermolysis bullosa, Rett syndrome, Love syndrome, Dowling-Meara. We have, on average, an index patient returns 51% of true positive patients in the top 30 most similar patients. It depends, in fact, of the number of patients, of the rarity of the disease, obviously, and how many concepts we have for each patient. If it's just one consultation, if we have an hospitalization of the patients. And the sensitivity is on average 20%. We published our result in the Journal of Biomedical Informatics. And just for the little story, a short story, when we evaluated the ability of diagnosed APDS, we use the APDS cohort. We keep one patient from the cohort as an index patient. We put all the other patients in the data warehouse, and the aim of the game is to be able to have in the top 30 patients, top 20 patients with APDS syndrome. And when we did the evaluation with each time a new index patient from the cohort, we have a false positive patient, always the same. And false positive because he was not in the cohort at the beginning. And if we look closer to this patient, his last visit was in 2011, and he was unclassified immune deficiency. We know that the APDS, the two genes are responsible for this syndrome, PIK3CD and PIK3R1, discovered in 2013 and 2014. So I asked clinicians of Necker Hospital to look closer to this patient, and they found a new deletion on this gene. So we diagnosed a patient during the evaluation of this clinical decision support system. So it was a success. We integrate the similarity algorithm in Dr. Warehouse, so now the clinician can use it to both way. We have a new mutation in the patient, we want to find similar patient with the same mutation. Or we have an undiagnosed patient, and maybe in the similar patient, we have diagnosed patient with new clue to diagnose our patient. Another use case is Ask Dr. Warehouse. So the idea this time is quite similar, but based on the data automatically extracted from the patient records of Necker Hospital, I want to have a proposition of disease or gene. So you just put a query, alopecia and immunodeficiency and diarrhea, for example. It will find the patients with the disease, and it will aggregate all the information it can find in the narrative record of those patients, and he compute a score of association, and you have the top 30 concepts associated to those patients. And the first one is TTC7A. Our system said that it's very associated to alopecia, immunodeficiency, and diarrhea. Another example is immunodeficiency, hyper-IgM, and bronchial dilatation. And the first one is APDS, and you have PIK3CD at the 5th position. Yeah, you don't have PIK3R1, I don't know why. So the idea is to have an architecture oriented toward artificial intelligence. You have all data available in the hospital. You have tools to recruit the patients, to extract the data, to enrich the data with HPO or other thesaurus. So you can develop your machine learning methods with mathematical artificial intelligence. You have to evaluate the CDSS on data, and you can apply the new CDSS, the new algorithm, on the data of the hospital, and the clinician can use it on the patient's data, not the patient's really. We published Dr. Warehouse in 2018, and Dr. Warehouse is open source now. From the beginning, I talked only about clinical sign, clinical phenotypes, and just gene mutation. And the next step now is to work on omics data. So we have a new project led by Frédéric Rioloca. This is a 5-year project started this year, a little bit late due to COVID-19. And the idea is to put all the omics information in addition to the clinical data, to have a new stratification of patients. We will use network inference and artificial intelligence to compute all this data together. And the idea is to have a personalized treatment to validate biomarkers and to build an atlas of molecular interactions driving autoimmunity and autoinflammation. The ultimate goal is to build a new software for guiding clinicians about these PIDs with autoimmunity. Thank you for your attention. I especially want to thank all the reference center for rare disease in Necker Hospital, the Necker Clinical Department with Felipe Suarez, Olivia Boyer, and all the imaging research teams like Sven Kraker or Geneviève de Saint-Basile. Thank you.
First of all, I would like to thank the organizers for giving me the opportunity to present our work. I have no conflict of interest to disclose. The crosstalk among thymic epithelial cells and developing thymocytes is fundamental for the correct development and selection of T lymphocytes. Because of this, mutations affecting either thymic epithelial cells or thymocytes' development and/or function will both result in T-cell immunodeficiency. For example, DiGeorge syndrome is caused by defect in TEC development, while RAG defects affect T-cell development, but both diseases present with T-cell immunodeficiency. For this reason, determining whether the T-cell lymphopenia is caused by a hematopoietic intrinsic or extrinsic defect in a timely manner is critical in order to decide the most appropriate treatment for the patients, namely bone marrow transplant versus thymus transplant, respectively. Recently, a new powerful technique to model in vitro T-cell differentiation has been published by the group of Gay Crooks and Amélie Montelhagen. This new technique involves the generation of artificial thymic organoids, or ATOs, by aggregating CD34 positive cells isolated from mobilized peripheral blood or bone marrow with a murine stromal cell line engineered to express the DLL1 or DLL4, the notch ligands, and it's called MS5 DLL1 or DLL4. The aggregates are then plated on a membrane floating on a medium, which is based on RPMI, is serum-free and supplemented with B27 supplement, IL-7, free 3 ligand, and ascorbic acid. Full T-cell maturation can be achieved after 5-7 weeks of cultures of the ATO. We use the ATO system to evaluate in vitro T-cell differentiations from CD34 positive cells obtained from bone marrow or mobilized peripheral bloods of patients carrying known defects affecting hematopoietic cells or thymus. Using the ATO systems, we were able to reliably determine the hematopoietic intrinsic or extrinsic nature of the defect and define the exact stages of T-cell differentiation affected in the patients with hematopoietic defects. Among the patients that we studied with this system, there are SCID patients and patients with reticular dysgenesis. Both types of patients present a very early block in T-cell differentiation at pro-T stage and pre-T1 stages, respectively, in addition to a survival defect. We then studied also patients with RAG1 or RAG2 gene defect, and this type of patients showed a much later block in T-cell differentiation and double positive stage, differently from what predicted in mouse models. Finally, we studied also patients with DiGeorge syndrome, and this patient showed no block in T-cell differentiation as expected because of the hematopoietic extrinsic nature of the genetic defect carried by DiGeorge syndrome patients. We thus believe that the ATO system can be used to determine the hematopoietic intrinsic or extrinsic nature of the defect in T-cell immunodeficiencies of unknown causes. Among the patients that we studied using the ATO system, we also evaluated patients carrying Icaros mutations. Icaros is an essential transcription factor expressed throughout hematopoiesis that plays a role in lymphocyte and myeloid differentiation and negative regulation of cell proliferation. Somatic mutations in Icaros have been linked to the development of B-cell acute lymphoblastic leukemia in children and adults. More recently, heterozygous germline mutations in Icaros have been characterized and give rise to distinct clinical phenotypes based on the nature of the mutations. Indeed, patients carrying mutations causing Icaros hypolinsufficiency present with common variable immunodeficiency associated with B-cell immune deficiency, BLL susceptibility, and autoimmune manifestations. No clinical T-cell defects are evident among these patients. Instead, patients carrying dominant negative mutations present with combined immunodeficiency characterized by a strong T-cell defect affecting activation, signaling, proliferation, T-helper generation, and memory generation. When we studied CD34 positive cells from Icaros patients in the ATO system, we observed in patients with both types of mutations a similar block in T-cell development at the stage of CD5 positive, CD1A positive, CD7, resulting in a decreased progression to the following stage of CD1A positive, CD7 negative stage, which represent the CD4, CD8 double positive stage, as shown in the red boxes. The block was more severe in the patient with a dominant negative mutation, as you can see here. In fact, the patient with haploinsufficiency in Icaros could progress beyond the block at CD CD7 positive, CD1A positive stage, and could generate some CD4, CD8 double positive cells that led to the final generation or residual amount of CD3 positive T cell alpha beta positive mature T cells, as shown in the green boxes. Instead, the patient with a dominant negative mutation had virtually no mature T cells produced. The defect in differentiation in Icarus patients was also confirmed when we look at the absolute numbers. And in fact, both patients showed a decreased number of double positive cells and decreased number of mature T cell alpha beta CD3 positive cells. Interestingly, in addition to the block at CD1A positive, CD7 positive stage, only the patient carrying the dominant negative mutation showed a peculiar block at the CD4 negative, CD8 alpha positive stage, shown in the red boxes, which is in normal T cell development only a transient stage in T cell differentiation. In conclusion, using the ATO system, we demonstrated that both types of Icarus germline heterozygous mutations cause a block in T cell differentiation at the stage of CD5 positive, CD1A positive, CD7 positive, preceding the stage of CD4, CD8 double positive. The patient carrying the haploinsufficiency mutation in Icarus shows a leaky block that allows for a reduced number of T cells to reach full maturation, and that could account for the mild T cell defects found in these patients. Instead, the patients carrying the dominant negative mutation shows also peculiar accumulation of CD4 negative, CD8 alpha positive cells, suggesting a role for Icarus in the upregulation of CD8 beta. Further studies of gene expression on cells isolated from the ATOs are planned to assess the pathways affected by Icarus mutations in order to clarify the precise role of Icarus in human T cell development. Finally, I would like to acknowledge all the people involved in these studies. First of all, people in Dr. Notarangelo's lab at NIH, who helped in the ATO generation and culture, in particular Francesca Pala, Amelie Montelhagen and Gay Crooks from UCLA for sharing the ATO technique, all the collaborators and physicians who share the patient cells with us, in particular Hezun Kuhn and Sergio Rosenzweig for the Icarus patients, Dr. Notarangelo for allowing me to work on this project, and all of you for your attention. I'd be happy to take any questions.
Thank you for inviting me to present those data. That is a new test to assess the functional defect in SCID patients without molecular diagnosis. I have nothing to disclose. And we know that we can have a dilemma in diagnosing patients with unclassified SCID. You know that in North America, patients are mostly diagnosed with newborn screening. And around 5 to 10% of those patients lack identification of the genetic diagnosis. And when the phenotype is particularly T minus, B plus, NK plus, then it can either be an intrinsic hematopoietic defect or a THYMIC defect. And for the clinicians, it's difficult to know how to treat those patients and to go straight forward to a bone marrow transplantation, while maybe it is a thymus transplantation that is needed. So there is clearly an unmet clinical need, and we need a simple test to differentiate those patients. So how to do that? We need CD34 positive cells, so it could be bone marrow or peripheral blood. For sure, peripheral blood would be better. It has to work with low CD34 numbers, because with peripheral blood in newborn, we can have a lot. And it needs to be not too long. Some weeks it's okay, but over it could be complicated for the clinicians. So it would be great if it's also feasible with CD34 positive cells from frozen PBMC, because sometimes patients have frozen PBMC and it's complicated to reharvest them. So when you look at the T cell differentiation in OP9DL4 monolayer, what I mean is the classical way of doing it in a culture of monolayer, we see that although the cord blood works very well, in 3 weeks you can see double positive CD4, CD8. Peripheral blood doesn't work with 2,500 CD34 positive cells, and usually it's 5,000. Just by reducing 1/2, it doesn't work. And at 5 weeks, you see that cord blood, it's perfect, but peripheral blood doesn't work. So we changed the system and we try to increase our test. We used OP9DL4. We co-culture with OP9DL4 in 3 days instead of monolayer, because it was shown to be better in a publication by the Gay Crooks group. We optimized the media composition which contains serum, and we selected porous culture insert that could not spread the liquid. So that's the thing we set up, and try to see if it works for T-cell differentiation. Here you see what it looks like. You have the insert, and here you can see the very tiny organoid that we made. So when we do that and we compare cord blood with peripheral blood, we see that it's okay, it works at 3 weeks. And at 5 weeks, you see the difference here with 2,500. When you compare with the previous slide I showed you, it's relatively robust and it works well. So we said, okay, now let's test for THYMIC deficiency, and the differentiation should be normal. Intrinsic hematopoietic should lead to a normal, and we also wanted to test OMEN and ADA deficiency. So we looked at first IL-2 gamma-C SCID. So here is the control with a perfect differentiation in CD1A, CD7, CD4, CD8, and T cells also that are here. And in gamma-C, you see that it's completely blocked. Actually, there is an increase in CD7 cells, no CD4, CD8, and absolutely no T cells. So there is a complete block in the differentiation. When we look at RAG typical SCID with no T cells, again, the control is here. And we see that although the CD7, C1a is okay, we don't have the differentiation in double positive cells, and we don't have T cells. And when we look at OMEN syndrome, patients with a RAG mutation also, we see here the double positive, but we don't see the CD3 positive. It's interesting to know that Gigi Notarangelo lab, Marita Bosticardo, they used the same sample as this one, and they had a different result with bone marrow. But it may be that their culture was shorter, because when the culture they last for 6 weeks, then they observed a decrease in those double positive cells. So the kinetic of the culture may be important. So gamma-C, it works. RAG, it works. What about DGEORGE? So here we have a patient with DGEORGE, and you see that as expected, the differentiation is completely normal, double positive, CD3 positive cells, and double positive for CD7, CVD1A. And when we had also a patient with a TBX1 mutation, we found exactly the same thing, normal, meaning that for a THYMIC deficiency, we have a normal differentiation. Then we looked at ADA deficiency, and it should be abnormal. And the surprise is that here it's normal, meaning that it's the limitation of our test. Maybe there is a transcorrection of the ADA by either the feeder cells or by the serum, or maybe because of the absence of toxicity that has not accumulated in the thymus in vivo. Here we are in vitro. But what it means is that those metabolic deficiency cannot be diagnosed with our technique, but usually they are diagnosed otherwise. So we assessed also a real case, a patient from California. We could get 5 milliliter blood. We could isolate less than 500 CD34 positive cells, and we observed that the patient had a complete block of the CD4, CD8 positive, double positive cells, unlike the control. And the patient went to a bone marrow transplantation and is now doing well, meaning that our test helped the clinician to decide what to do. So in conclusion, here is the graphical abstract that we see here. If we have an unknown cause, we take 5 milliliter blood, we isolate the CD34 positive cell, we do our 3-day culture with an artificial thymus in a liquid saline interface. If it's normal, it's likely a thymus transplantation that is needed, and if it's abnormal, it's likely a bone marrow transplantation. And here you have the different results of the data. But the thing is that when it's completely abnormal, the therapeutic implication is relatively easy. But when it's normal, it may be a little bit complicated. Recently, we had a patient, and in vitro, we could differentiate some T cells. But when we looked at the markers, it was completely abnormal. And the patient had the presence of CD34 positive cells at the surface of the cells that last for weeks in the culture. And together with the same exam in the bone marrow in the Marita Bosticardo lab, we decided that we compared our data, and we say that although when we push the system with T cells, this patient had really abnormal differentiation for the bone marrow cells, suggesting strongly an intrinsic deficit. So when it's completely abnormal, it's relatively easy. When it's normal, it takes more attention to be sure that the differentiation is completely normal. So I thank you for your attention, and I wanted also to acknowledge all the people involved in this work. So first in my lab, Panos Bivsha, who had the ingenuity to create all those things on this artificial THYMIC organoid way of culturing cells, and helped by Aurélien Colomartino, PhD student in my lab. Jane Lyding, Sun Young Pai, Jennifer Puck, Louise Marquette, Nicolas Hartog, and Joe Church, who provided samples, and with the help of Gigi Notangelo and Marita Bosticardo at the NIH, who worked on some and who shared some samples and with whom we compared the data because they used the kind of same test with bone marrow cells. So thank you again for your attention.
Hello, I am Eline Beyls and I'm a PhD student at the Radiobiology Research Group at Kent University. Today I want to present you my recent work regarding the radiosensitivity assessment of fibroblasts from patients with a primary immunodeficiency. The questions can be submitted during the talk and will be answered after the session. I have nothing to disclose. You all know that primary immunodeficiencies are a heterogeneous group of genetic disorders. More than 400 genes are involved in PID, and even a single gene defect may result in a variable disease presentation. But establishing a genetic diagnosis is important. We can all agree that the genetic diagnosis is important as it leads to better healthcare for the patients. Besides this problem, there is also a small subgroup of PIDs where the immunodeficiency is related to a DNA repair defect. Patients with such a DNA repair disorders are characterized by a pronounced radiosensitive phenotype. So we aimed at offering radiosensitivity analysis for these patients. This will be an added value to guide the genetic screening, but also identifying radiosensitive individuals is important to limit the exposure to DNA damaging agents. So this radiosensitivity analysis is important to both optimize the diagnosis and the therapeutic management. At the beginning of the presentation, I mentioned that the cell type we are using is are fibroblasts, but why would we do this on fibroblasts? It's indeed true that routine radiosensitivity analysis is performed on peripheral blood lymphocytes. But for SCID patients, this is not an ideal cell type. SCID patients often have very few patient-owned lymphocytes or even none at all after transplantation. But the radiosensitivity analysis is really important for the SCID patients, as some SCIDs have a defect in the non-homologous end joining pathway, and this is the major pathway responsible for DNA double-strand break repair. So radiosensitivity analysis is really important to be able to perform it for SCID patients. And fibroblasts isolated from skin biopsies can be a feasible alternative for the lymphocytes. So therefore, the aim of this study was to develop a reliable radiosensitivity assay on fibroblasts. The assay we used is a G0 micronucleus assay. With this assay, the DNA repair capacity is used as an endpoint to estimate the individual radiosensitivity. We start from a small skin biopsy from the patient, where we then isolate fibroblast cultures from. We then irradiate our fibroblasts in the G0 phase of the cell cycle. It is important to irradiate them on synchronized cells in the G0 phase, as the non-homologous end joining pathway is mainly active in the G0 phase of the cell cycle. Then the micronucleus assay itself will take 4 days, and after that we analyze our cells manually by scoring on the microscope. In a first step, we analyzed fibroblasts from healthy controls. Here on the y-axis, you can see the number of micronuclei. You can clearly see on the graph that for all 3 cell lines from healthy individuals, that the obtained results for micronuclei are very similar. And also within one cell line, the variation is highly acceptable. Then we looked at DNA repair defective cell lines. We first analyzed 2 ATM deficient cell lines who are known to be extremely radiosensitive. The criterion for radiosensitivity that is commonly used is that patient values need to be higher than the mean of the healthy controls plus 3 standard deviations. And here on the graph, you can clearly see that the obtained results for micronuclia for these 2 ATM deficient cell lines are higher than the curve for 3 standard deviations. So this newly developed assay, the expected radiosensitivity is confirmed. And we could also obtain 3 cell lines from SCID patients. The first 2 are 2 Artemis deficient cell lines. Artemis deficiency leads to a SCID phenotype, and Artemis is also involved in DNA repair. So you can see on the graph that indeed values for micronuclei are much higher than the 3 standard deviations curve. And this is in contrast to the RAC2 cell line. RAC2 deficiency leads to a SCID phenotype, but RAC2 is not involved in DNA repair. And here you can see that indeed on the graph that the values for micronuclei are similar to those obtained from the healthy controls. So RAC2 deficiency shows a normal radiosensitivity. To conclude, with this newly developed micronucleus assay, we could confirm the suspected radiosensitivity for the analyzed cell lines. And as last, we are looking for patient samples for further validation. So if you have SCID patients that are eligible for radiosensitivity analysis, do not hesitate to contact us. And finally, I want to thank our radiobiology research group, especially my supervisors, for giving me the opportunity to conduct this research. Thank you.
So hi, my name's Alex. I'm doing a PhD at UCL, and today I'm going to be talking about some computational modelling that we've been doing on STAT1 gain-of-function mutations. So I've got to disclose. So STAT1 is a transcription factor, and it functions predominantly in an immune context downstream of critical cytokines such as interferons, which I'm showing you here. So under inactive status, STAT1 mainly exists in a dimer, a homo or a heterodimer format, and upon ligand binding to cognate receptor, these dimers then dock at the receptor site, where they're then phosphorylated by JAK proteins before they translocate into the nucleus, either alone or in complex with other proteins, where they then bind to specific DNA promoter elements and induce the transcription of downstream genes. They are then dephosphorylated before they exit to the nucleus to undergo successive rounds of signaling if applicable. So to date, there are 112 different mutations, gain-of-function mutations. These are congenital, they're heterozygous, and they're now known to be found throughout all the domains of STAT1, but predominantly within the coiled coil or the DNA binding domains. So on a molecular level, the immune cells of patients seem to exhibit enhanced levels of STAT1 phosphorylation upon stimulation with a STAT1 inducing cytokine. And this is what I'm showing you on the top row here. So for example, if you look at the gain-of-function patient, you can see from the fold change from unstimulated to stimulated is almost double that of the healthy control. And as a consequence, this leads to greater expression of STAT1 inducible genes. An example of this is shown at the bottom here with CXCL10, as you can see upon stimulation. Now, this translates to an extremely varied clinical phenotype, and they mainly present with what is known as chronic mucocutaneous candidiasis. And this is a broad infectious manifestation of severe T-cell immunodeficiency. And it mainly involves recurrent or persistent infections of the skin, the nails, or the genital mucosa via Candida species. But they can also present with a whole host of infectious disease, as well as autoimmunity, and an increased propensity to both aneurysms and cancer. So the molecular mechanism is still poorly understood. There's been a few hypotheses, but the one favoured seems to be delayed dephosphorylation due to the destabilisation of the antiparallel dimer, because this is thought to be the only conformation which presents the phosphotyrosine residues accessible to a dephosphatase enzyme. But there's been a few contradictory reports to date, so through computational modelling, we wanted to investigate this further and see what is actually happening at the structural level. So under inactive status, STAT1 homodimers mainly reside in what is deemed an antiparallel conformation, in which the coiled coil and the DNA binding domains are interfacing with one another. But they can also reside in what is known as a parallel via NTD conformation, in which the N-terminal domains are interfacing with one another and the nuclear localization sequence is exposed. But following phosphorylation, i.e. activation, there's a high affinity for the parallel via CTD conformation, in which the C-terminal domains are interfacing via respective SH2 and phosphotyrosine residues. And when we mapped each gain-of-function mutation onto these conformations, so each sphere here depicts one mutant residue, we found that groups tend to line the interface, or the dimer DNA interface. So we decided to explore this further by generating the following schematics, an example of which is shown on the right here. Now, these convey a lot of information, but for the basis of this talk, they're showing what residues are interacting with one another in a given dimer conformation. So each half circle that you can see, that depicts one monomer. And the grey lines that you can see, they're showing the residues which are interacting with one another in a given dimer conformation, in this case, the antiparallel dimer. And the target residue for this example is that of threonine 385, which is located by these green asterisks here. And the black lines that you can see, this is showing the residues which threonine 385 is interacting with. So you can see it's having a few local interactions, but more importantly, it's interacting with the residues of the opposing monomer, suggesting that it's making up this vital connection leading to the adoption of the formation of the antiparallel dimer. Now, we generated these for every STAT1 gain-of-function mutant, and I'll take you through a couple of examples in the following slides, and it's these black lines that I want you to look out for. So on the top, we're looking at the interactions of D165, which is mutated to G or a H conferring gain of function. And you can see that this is playing a particularly prominent role in the adoption of the antiparallel dimer. So you can see the residues interacting with the opposing monomer to essentially make up this antiparallel dimer. But you can see it doesn't play a role in the adoption of the NTD or the CTD dimer. Now, on the bottom row, you can see there are 70 interactions, and this is mutated to CH or P, resulting in gain of function. And you can see this doesn't play a role in the adoption of the antiparallel nor the CTD, but plays a particularly prominent role in the formation of the NTD dimer. Now, on the top, you can see the V653 interactions, which are commonly mutated to isoleucine to confer gain of function. And you can see this is only playing a prominent role in the adoption of the CTD dimer. So from these schematics, we were able to determine that the residues which are being mutated to confer gain of function, they seem to play an active role in the interfaces of one or more conformations, or they interact with the residues that do. And just to control for this, we also mapped the whole load of neutral mutations, as you can see on the bottom row here, and we found that these tend not to interact with the opposing monomer in any way, nor do they interact with the residues that do. So going in further, we wanted to look at these interfaces in a bit more detail. So I'm showing you two examples of this here. So on the left, you can see the antiparallel conformation, and in particular, the T385M mutation. So you can see T385 is interacting with D168 on the opposing monomer via a polar contact, which is depicted by this dotted yellow line here. You can see when we mutate this to methionine, this interaction is lost. So therefore, we think this is destabilizing the antiparallel conformation. And on the right-hand side here, you can see that we're looking at the CTD conformation, and in particular, the T419R mutation. You can see T419 here. And when we mutate this to arginine, you can see the residue now inserts itself much deeper into this pocket of DNA. And because arginine is positively charged at physiological levels, as shown by the blue color, we think this is having a stronger interaction with DNA, resulting in prolonged DNA binding and therefore enhanced expression of STAT1-mediated genes. So to conclude, we think we provided evidence to suggest that gain-of-function mutants are affecting one or more dimer interfaces, and they can do so directly or indirectly. We've also provided evidence to support the destabilization of the antiparallel dimer hypothesis, and this can result in delayed dephosphorylation, or maybe it could result in we think it might result in the adoption of a parallel via NTD-like conformation, which exposes the nuclear localization sequence, therefore leading to STAT1 expression of STAT1-mediated genes under inactive status. For those that line their NTD dimer, we think this is leading to premature nuclear entry, similar to what I've explained here. And for those that are lining the CTD dimer or the CTD DNA interaction, we think they're causing prolonged DNA binding. And finally, in the context of diagnostics, we're currently generating these circular schematics for each STAT1 residue, because we think this might be able to speed up the diagnosis process, providing, of course, that functional validation proves successful. So we think if you're to come across a variant of unknown significance, instead of waiting weeks for the functional validation to come back, you can quickly scan these images, see if it lines the interface, and determine if it's likely to have an effect or not. And so thank you for listening, and thank you to everyone involved, particularly our collaborators out in Japan, Darren and Sol-Ling, and I welcome any questions.
Good morning, I'm Marta Bustaffa from Garcelon in Genoa. Any question or suggestion are welcome through the Q&A function in the top right corner of the screen. I'm going to present you a case report of autosomic recessive CGD due to P40 deficiency. These are my disclosure. Deficiency of P40 has been recently described in 25 patients with chronic granulomatous disease with atypical manifestation. Patients with deficiency of P40 differs from classical CGD in many aspects. They are less prone to fungal infection and mortality is significantly lower in this group of patients. Clinical presentation is characterized by hyperinflammation and peripheral infection, especially skin and ocular infection. Moreover, diagnostic can be challenging because DHR oxidation test is reported to be normal or slightly reduced after PMA stimulation, unlike classic CGD. This is a report of a patient with P40 deficiency with prevalent skin manifestation. She is a girl born from non-consanguineous Sri Lankan parents. In 2009, when she was 5 years old, she was referred to our unit for recurrent skin infection, initially interpreted as atypical psoriasis. Psoriasis was resistant to usual treatment, but responsive to antibiotics. Repeated skin culture revealed the cutaneous infection by Pseudomonas aeruginosa and Staphylococcus aureus. These findings led our colleague to suspect a primary immunodeficiency, and the patient was referred to our unit. No abnormalities of lymphocyte number and function were detected at the diagnostic workup. Oxidative burst measured by DHR test resulted modestly impaired, and in the suspect of CGD, prophylaxis with cotrimoxazole was started with improvement of skin lesion. That was one of the first DHR tests done in our lab. So the DHR was repeated in another center for confirmation, but resulted normal. So the diagnosis of CGD was questioned, and unfortunately, the patient was lost to follow-up and discontinued the treatment before genetic analysis could be planned and performed. 11 years later, she came back to our attention because of keratoconjunctivitis responsive to vancomycin, severe erythematodesquamative dermatitis, soft tissue hypertrophy with probably granulomas, and hidradenitis suppurativa. So we performed another DHR test that was repeated and resulted markedly reduced following a PMA stimulation and absent following E. coli stimulation. Genetic analysis revealed an R105Q homozygous mutation in NCF4 gene. So the diagnosis of autosomic recessive CGD due to P40 deficiency was posed. Here you can see the DHR results of our patients in dark blue, compared with 2 patients with X-linked CGD in green and 2 autosomic recessive CGD in lighter blue, with 2 healthy control in red. That's interesting because in the previous report in the literature, oxidative burst was reported normal or slightly reduced after PMA in P40 deficient patients, while in our patients, oxidative burst was markedly reduced after PMA and also absent after E. coli stimulation. Our patient antimycotic and antibacterial prophylaxis was started with some improvement. Steroids were necessary to control the severe skin manifestation with an improvement of dermatitis and slight improvement of hidradenitis suppurativa. Radiological and microbiological tests did not reveal invasive fungal or bacterial infection, and no gastrointestinal inflammatory involvement was proven. Bone marrow transplantation was discussed, but finally not considered in these patients since the mild phenotype. In conclusion, until now there are a few cases described with this mutation in literature. As this case report shows, diagnosis of P40FOX deficiency might be a challenge. In this formal CGD, DHR might be misleading because of a reduced but not absent oxidative function. Moreover, classic CGD-associated infections are not reported in these patients, while skin or ocular infections are quite frequent. And finally, P40FOX deficiency is associated with two prevalent inflammatory manifestations. In the previous report, inflammatory manifestations were mostly pulmonary or gastrointestinal, while our patient has a marked subcutaneous inflammatory involvement. Thank you for your attention.
Good afternoon, everybody. My name is Maartje Blom, and I'm a researcher at the Leiden University Medical Center in the Netherlands. And today I would like to discuss with you the role of epigenetic immune cell counting as a second-tier test in newborn screening for SCID, or severe combined immunodeficiency. I have nothing to disclose. So as you all know, newborn screening for SCID is based on the detection of T-cell receptor excision circles. And TREX are actually a marker for THYMIC outputs, and that is why newborn screening for SCID is accompanied by many secondary findings and also false positive cases. For example, if you look at the data of the SONET study, which is a Dutch newborn screening pilot for SCID, we screened over 140,000 newborns, and we found 43 preterms with low TREC levels that require a second heel prick. And also we had 47 referrals, which included one SCID patient, a lot of secondary findings, and also 5 false positive cases. And every referral is actually associated with a lot of feelings of anxiety and insecurity for parents. So the emotional impact of a referral is very high. That is why we believe there is need for a second-tier test after TREC analysis in order to reduce the number of secondary findings and, more importantly, false positive cases, but also to increase the sensitivity for newborn screening for SCID. So today I want to talk to you about epigenetic immune cell counting on dried blood spots. So epigenetic immune cell counting is a technique that was first described by Barron et al. And it uses unique DNA fragments that are actually not methylated in your target cell types, but they are methylated in your non-target cell types. So if you look to the left, you have your target cell type with a unique not methylated fragment of DNA. And if you will now add bisulfite conversion, your unmethylated cytosines will be converted into a uracil. However, in your non-target cell types, your cytosines are protected by the methyl group and will not be converted. You can now develop specific primers for your converted DNA, and this way you can measure with qPCR the different types of leukocytes. So we analyzed a relatively large group of original newborn screening cards that included healthy newborns, PIT patients, preterm newborns with low TRECs, and also referrals with low TRECs. And the protocol was actually very standard, including DNA extraction and lysis, a bisulfite conversion step and purification, followed by qPCR amplification and measuring. And we can measure the relative cell counts of different kinds of leukocytes, including regulatory T cells, B cells, and K cells and granulocytes. But for now, we will focus on our CD3 positive T cells. So first, let's look at the correlation between TRECs and your unmethylated CD3 copies as measured with qPCR. And we will look at the entire study population together. We found a correlation of 0.7, which is actually a strong correlation between TRECs and unmethylated CD3 copies. However, the correlation is not perfect, implying that epigenetic qPCR can actually produce some different results, which is also important when you have a second-tier test. So now let's look at the 3 study groups that we analyzed, first starting with our PIT patients. So we included a variety of PIT patients with differing diagnoses, not all with a T-cell deficiency. Let's focus on our SCID patients that are here in red. So SCID patients, of course, have 0 TREX and can easily be distinguished from our healthy controls. However, if we look at our unmethylated CD3 copies in the middle or our relative C-cell counts on the right, we can also see that they're absent for SCID patients as well, implying that epigenetic qPCR actually would function just as well as a TREC assay in identifying SCID patients in right blood spots, especially for these 4 SCID patients included in the study. So when we look at the preterm newborns, these were of course selected based on their low TREC levels. So of course there's a significant difference between our preterms and the healthy controls. So if we look now at the middle and the right blood, we can also see that they have significantly lower unmethylated CD3 copies and relative T-cell counts. However, there's some overlap between the preterms and the healthy controls, implying that if you would choose the right cutoff value, you could have prevented some additional second heel pricks for these preterms. So then the most interesting group of all are referrals. We included a variety of referrals with 6 false positive cases in green. And of course, on the left, all of our referrals have low TREC levels as they were chosen based on their low TREC levels and referred on their low TREC levels. But if you look at your unmethylated CD3 copies or at your relative T-cell counts, you can actually see that there's quite some overlap with your healthy controls, especially for the false positive cases in green, which are very overlapping with your healthy population. And this implies that if you would use epigenetic qPCR as a second-tier test, you could actually have prevented these false positive referrals. And with that, you could have prevented a lot of anxiety and insecurity for parents. So finally, we have one group of interesting infants left. We have 26 newborns with cards that were spotted with peripheral blood used for flow cytometry. So now we have blood in which we can measure TREX, unmethylated CD3 copies, and also we have the absolute T-cell counts measured with flow cytometry. And if you look at the left, you can actually see that there's only a moderate correlation with a correlation coefficient of 0.45 between TREX and your absolute T-cell count. While looking at the right plot, you can see that there's very strong correlation between your unmethylated CD3 copies and your absolute T-cell counts, implying that epigenetic qPCR is a more direct and also accurate measure for your T-cells. So this brings us to the conclusion and discussion. So epigenetic qPCR has the potential to increase your positive predictive value and sensitivity of newborn screening for SCID. It could help in the reduction of second heel pricks if that is incorporated in your screening algorithm, and also in a reduction in referrals, especially for the false positive cases. However, before this assay can be implemented in a screening laboratory, further development and optimization would be required. In addition to that, further evaluation of your epigenetic immune cell counting in the newborn screening field would be interesting as well. Could it serve as a first tier for other diseases, for example, looking at XLA and B cells, or IPEX syndrome and regulatory T cells, and of course also at severe congenital neutropenia. Finally, it's also important to explore other potential second-tier options, for example, next-generation sequencing. And with that, I would like to end my presentation. I would like to give a special thanks to Dr. Mirjam van der Burg and also to everybody at APImmune for the great collaboration on this project. And I would like to thank you for your attention.